I hope it doesn't run out of it. Okay, so today we're going to do genetics. You've got your homework, page 7 through 14. Is what you're going to turn in online. You do have a quiz this week as well. Your final lab report is due next week, so it's going to be after you come back from spring break. So for your class, it's going to be Monday. You can still come in for lab and not have turned your lab report in by then, okay? So what you will have is up until 11:59 on Monday night in order to submit that. When you go to submit your lab report, there is a program called Safe Assign. When you go to submit that, you're going to submit your report, hopefully, in a Word document. Make sure that it is compatible with Blackboard and Safe Assign, or else it will not be able to run it through that program. That is a requirement for submitting your lab report. So when you scroll down to the bottom of it, you're going to have one little checkbox, and it says, allow Safe Assign to run through your lab report. You have to check that box. If you don't check that box, it won't come to me on my end where I can grade it. All right? I'm going to set unlimited attempts. Okay? If you upload it and you're like, oh, I forgot to include my reference sheet, and you need to upload it again, that's fine. I will grade the last submission. All right? Do not wait till 11:58. Just in case Blackboard is down. And then you can't submit it. Okay? Alright, so we're going to talk about genetics today. We're going to talk about some terms that will be very important in order for you to answer questions right when it comes to quizzes and final practicals. Alright, we're going to talk about dominant. We're going to talk about recessive. We're going to talk about heterozygous and homozygous. We're going to do a Punnett square. We're going to do monohybrid, and we're going to do a dihybrid. At the very end, some home activities that you're going to include, that you're going to work on, is going to include some DNA and RNA. I do have a couple copies of the lab. Does anybody need it? It's about 14 pages. You guys go ahead and come on up and get it if you need it. Okay, so when we talk about Mendel genetics, the study of genetics is how we carry on genetic material to our offspring. The possibility of heredity, and whether we're going to have dominant genes, whether we're going to have recessive genes. All right. So each individual inherits one set of chromosomes from each parent. We already know that because we talked about mitosis, right? Mitosis was just for repair and growth. Meiosis was going to be for sexual reproduction. So we already know that we have 46 pairs of chromosomes, or 23. So we're going to get 23 from mom, 23 from dad. That's going to bring us back to 46. All right. Now, a gene codes for a protein. Building blocks for proteins are what? What are our building blocks for proteins? Amino acids. Okay? Amino acids are going to build for our proteins. The different versions of these genes are going to be called alleles. Each individual inherits 2 alleles for a trait. We're going to talk about this. We're going to use the Punnett square in order to see what type of information is passed down from parent and parent, as far as mother and father. All right, now let's talk about the terminology. And like I said, this is a very important. All right. Now, if I'm looking at the actual genes, we're not going to physically see genes. We're not going to look in the mirror and see genes. That's something that we would have to go get genetic testing for. So when we're talking about a genotype, that is going to be the gene. When we're talking about the phenotype, think of the word physical. So when we think of phenotype, we can actually see what traits are displayed. Brown hair, blonde hair, green eyes, tall, freckles, widow's peak, attached earlobes, no attached earlobes. All those different traits that we can physically see. That's the difference between the word of genotype and phenotype. Alright, then we have heterozygous and homozygous. What does homo mean? Same. What does hetero mean? Different, that's right. Okay? And we already know the word zygote, right? Brand new individual, heterozygous, homozygous. These are the two alleles. Okay? Now, if we're talking about a dominant allele, let's use R. If it's dominant, it's going to be written as a capital letter. If it is recessive, it's going to be written as a lowercase r. Now, if I have a homozygous trait, that's going to tell us that I have two of the same. Right? Homozygous. Big R, big R. Or I could have little r, little r. They are the same. So they are considered homozygous. What if I had a heterozygous? I'm going to have one of each. Hetero means different. This is dominant, this is recessive, but they are the same. So we're going to use the word dominant or recessive as almost like an adjective to describe these. So when we're communicating with somebody as a dominant gene or a recessive gene, all right? If we have two capital R's, they are homozygous, but they are dominant. So I would describe these as a dominant allele. Dominant homozygous. Or you can say homozygous dominant. Either way, as long as you put the dominant in there. Now, if I go over here and I'm talking about the two little r's, I'm going to have homozygous. But what am I going to put to describe it? Recessive. So that would tell me I have recessive homozygous. If I have hetero, that tells me both of them are different. I don't have to use a descriptive term for a heterozygous because I know that they are different from one another. All right, now let's look at this example. Starts right here. In a pea plant, purple is going to give us a big pea. It's completely dominant over the little pea. If a pea plant has purple flowers, we know it has at least one capital P. But we cannot tell just by looking at it whether or not both alleles are big or if we've got one big one and one little one, which was heterozygous. So what we're saying here is if I am purple, my possibilities of my genotype are going to be capital P, and little p. Right? Because my purple is dominant here, my purple is dominant here, and he's going to trump the white. The only way I can come up white is if I'm little p, because I don't have a dominant p. So that tells me I'm going to be without purple, which is going to be white. All right, now, moving on down. We already talked about crossing over. Alternative forms of genes known as alleles account for variation. We talked about crossing over, giving us an exceptional amount of variation in offspring. All right, if the possible alleles of a gene different, one may be dominant, meaning that it would mask the phenotypic expression of the other allele. So here the genotype would be, tell me what the genotype would be if I've got two capital P's. Homozygous, dominant. What is this one going to be? A big P and a little p? Hetero. What if I have two little p's?
Homo and dominant or recessive.
Good. Homozygous, recessive. Okay? So, two alleles for a heritable trait segregate during meiosis, because we already talked about meiosis, usually independent of other traits. Dominant alleles mask all other phenotypes, known as recessive. Okay? Now, you told me the genotype. What if I ask you what the phenotype of these were? What would you tell me? What would this one be? Purple. Purple. White. So you see the difference in the question, a genotype versus a phenotype. Make sure you keep those straight in your brains. All right, let's do this. Freckles are dominant. Freckles are dominant. If I have freckles, what are my possibilities of a genotype? A homozygous dominant, which is going to be big F, big F. What if I had a heterozygous? A heterozygous would be big F and a little f. Would that still give me freckles? Yes. What are no freckles? Both would be little f's. I'm going to let you answer these questions. What is the genotype of a person who is homozygous dominant? What is my genotype for a homozygous dominant? Tell me. Genotype, it's going to be the letters.
Two capital S.
Homozygous dominant, two big F's. What is the genotype of a person who does not have freckles? Little f, little f. What? Oh, we got a different question here. What is the phenotype of the heterozygous? So first of all, we got to figure out what is the heterozygous. It's going to be a big F and a little f. Do I have a dominant F in there? Yes, I do. If we have a dominant F, we have freckles. So because it asks for the phenotype, I'm going to give the physical characteristic, not the genes. So what am I going to put here? Freckles. I'm going to physically display freckles. All right, here's another one. What is the phenotype of the homozygous recessive?
No freckles.
No freckles is right. None. What are the possible, we're going back to the genotype, for someone who has freckles? Obviously, dominant homozygous. What's the other possibility?
A big F and a little f.
Yep. Big F and a little f. Because here, two dominant, one dominant, he's going to trump my recessive. So I'm going to have freckles. Any questions on that? No? All right. Let's move on down. What you're going to do at home. We don't have PTC paper at home, but we do have it here. So I'm going to let you do it. All right? What you're going to do is you're going to go home and you're going to look at friends or family and you're going to determine whether they have freckles or not. and then you're going to list their possible genotypes, how many people you had in your group, and then you're going to list the percentage with that trait of freckles, okay? If we didn't have PTC paper at home, then we could do something like widow's peak. Widow's peak is dominant. It's where the hairline goes back. All right, now we're going to talk about a monohybrid. We have two different forms here, a monohybrid and a dihybrid. Right now we're going to focus on mono, which means one. A monohybrid cross involves one trait. The term hybrid classically refers to a mix. In genetics, this means that the two alleles are different, thus the organism is heterozygous. Here, it says in tobacco plants, that's what you see of these tiny little plants in your buckets, leads to chlorophyll production, and then it is going to make the plant green. So if you are given a letter on a quiz or a final practical, use the letter that's given to you. We're going to have a big C and a little c. Big C says, I'm going to be green. If I have a little c, no chlorophyll is going to be produced, so therefore I'm going to be white. Now we're going to go on down here and we're going to fill out our Punnett square. We have to have directions in order to know how to do this. It says, fill out the Punnett square below for the cross of the P generation. That is the parent generation. The F1 is the first generation of the parent. So that would be the first offspring of those two. Now it says the parent generation was heterozygous. So we're going to do this like a basic multiplication. We're going to put big C and a little c. Big C and a little c. And we're going to multiply those. So we're going to have big C, big C, big C, little c. Big C, little c, little c, little c. Everybody know how to do that? Your C at the top is going to go down in each row. Little c down here. Combine your big C with this one, and you're basically ending up with 4, which equals 25% each. So 25, 50, 75, 100. Now let's look at the question. Based on your Punnett square, what is the expected phenotypic ratio? So let's look at what our results are. We've got big C, big C, big C, little c, big C, little c, and then little c, little c. What is my percentage of purple? How many purples?
75%.
Absolutely. 75%. How many whites?
25.
25. Now you can write it like that. Or you can write 3-to-1. But please make sure you tell us 3 purple, 1 white. What if I reworded the question and I said, what are the genotypes? Big difference in the answer, isn't it? If I put genotype, I could say 1, dominant, homozygous, 2, heterozygous, 1, recessive homozygous. So it could actually be 1-to-1 ratio. What if it asked me what is just the percentages of whites? You just have to give me 25%. It all is going to be dependent upon what the question asks you. That is why it's so very important to know your terms of homozygous, phenotypic, genotypic, all of those terms. Alright, now what you're going to do in lab today is you're going to take either the picture that you have in the bucket, or you're going to take your little plate that's got a picture of the tobacco seedlings. Alright? And you're going to count how many greens you have and how many whites you have. Okay? We came up with a 75 to 25 ratio. So we came to a 3-to-1 conclusion of green to white. That's what our results were, according to our Punnett square. So, if we count these, do you think we're going to get similar results? By looking at the pictures, yeah, because I can see a lot more green than I do whites. Okay? So we're going to pair up, if you'd like. Everybody has their own bucket if you want to. And we're going to count the differences between greens and whites.